All right, let's start talking about. We talked a little bit about in chapter 11 about microbial control using physical and chemical controls. Now let's talk about how we control microbes once they get out of the environment and into our bodies so that we can actually treat them. So we're going to look at some interactions between the drugs and the microbes and the drugs and the host, and we're going to see how these chemotherapeutic agents will work throughout the system. A basic overview of what we're going to look at on this chapter is we're going to just kind of look at some of the principles of antimicrobial therapy as well as the drugs that get used and how there are interactions between the drug and the microbe as well as the drug and the host, and how do we select a particular antimicrobial drug so that we are getting the right one. You'll see that there are a number of different antimicrobial groups, everything from antibiotics that take care of bacteria all the way through antivirals. We're going to spend most of our time talking, though, about the antibiotics because that's what we are going to be exposed to most. We will discuss a bit about the antifungals, protozoals, helminths, and virals, but most of it's going to be spent on the antibiotics. The principle of antibiotic therapy is to find some sort of antimicrobial therapy that can be administered to an infected person that will destroy the infecting agent without harming the host or the host cells. This is what is called selective toxicity. In some of these antimicrobials, this is a very difficult thing to achieve. With bacteria, there are a number of different targets that are different between the eukaryotic human host and the prokaryotic microbe. But when we're talking about eukaryotic to eukaryotic organisms, like in fungal and protozoal infections, this selective toxicity is a little more difficult to administer. Chemotherapeutic agents are going to be drugs that we are going to use, and we're going to look at them in regard to their origin. Where did they come from? What is their range of effectiveness, and whether they are naturally produced, like we're going to see that certain antimicrobials are produced by plants, by other bacteria, by fungi, mold, those kinds of things, or are they chemically synthesized? Are these going to be made in the laboratory? If we look at this table, I don't believe this table is in your book anymore. This is one from a previous chapter, but I think well, yes, it is. I'm sorry, it is on page 328. This basically gives us some characteristics of the ideal antimicrobial drug. If you look, here is a list of things that range from selective toxicity to the price. Is it reasonably priced? Does it disrupt the host health system? All of these things, if we could have an antimicrobial that fit all of these, we would have a very significant drug. This table here is just kind of a dictionary for you. It's not one that you really have to memorize or anything like that, but as you read through the book and as we go through this lecture, this way you can have an idea of what we're talking about so that you can get an idea of the terminology of chemotherapy. When I say chemotherapy, I'm talking about any chemical that is used for a therapeutic purpose. Aspirin is chemotherapeutic. I'm not talking about the drugs that are used for cancer or anything like that. So this is basically chemotherapy, any kind of chemical that is used for treatment of some sort of an ailment. There are some different origins of antimicrobial drugs. The first antibiotics basically were identified as metabolic products of specific bacteria and fungi. The bacteria that are most common for the production of antibiotics are the Streptomyces genus and the Bacillus genus. These bacteria use these antimicrobials as a way to destroy the competitors in their environment. If they can produce this antibiotic, they can then kill all of these susceptible organisms that are in their environment, and it gives them a selective advantage. They have more food, they have more space, and they can grow and maintain their metabolism without a whole lot of interference from other bacteria, other populations. Certain molds, Penicillium and Cephalosporium, These are both types of molds that we get our penicillin and our cephalosporin drugs from. We also have some drugs that are created by altering the structure of these naturally occurring drugs. You have some what they call synthetic or semi-synthetic penicillins that have been changed up a bit to have some different characteristics that make them maybe a little more potent, or they work a little bit better. And so these are basically our synthetic drugs. We also have other scientists that basically are looking for metabolic compounds in other antimicrobial or in other species other than bacteria and fungi. Do we have some helminths or some parasitic worms that will release these antimicrobial substances that have an effect on other organisms? The goal of the antimicrobial drug is to essentially disrupt some sort of cellular process or structure of the bacteria, fungi, or protozoan, whatever we are trying to inhibit. Or it's trying to inhibit viral replication. And we're going to talk about a few antiviral drugs, and they have a multitude of different effects in how they stop the replication of viruses. Most of these interactions between drugs and microbes are going to act on the interference with the function of a number of enzymes that are required to synthesize or to build their large macromolecules like proteins, carbohydrates, lipids, or it will destroy a structure that is already within the cell, like the plasma membrane, or maybe it'll interfere with protein synthesis by attaching to the ribosome. So if they're going to interfere somehow with function or destroy structure. Drugs should be selectively toxic. If you're taking an antibiotic that ends up killing you, it didn't really do you a whole lot of good. But most of our drugs do a pretty good job of selective toxicity. They're going to kill the infecting organism without harming the host. There are 5 major mechanisms of actions or targets of the antibiotics. The first one are cell wall synthesis inhibitors. Then you have nucleic acid synthesis inhibitors, protein synthesis inhibitors, cell membrane structure or function interfere. They're going to interfere with these cell membrane structures or functions. Some of them will even interfere with the ability to build the cell membrane. And we also have the inhibition of folic acid synthesis, or a metabolism inhibitor. The first antimicrobial drugs that the first mechanism or target that I want to talk about are the cell wall synthesis inhibitors. Most actively replicating cells are constantly making new peptidoglycans so that they can keep that cell wall intact. What penicillins, for instance, and cephalosporins do is they are going to react with a number of enzymes that are required to cause to complete the structure of peptidoglycan. These are going to be bactericidal, meaning that they're going to kill the bacteria rather than inhibiting. If you look here at this picture, you'll see that we have a cell that has a good peptidoglycan cell wall. We're going to expose it to cephalosporin or to a penicillin, and we're going to see that this cell is going to begin to have some weak points within the cell. What happens is these are going to be areas that are going to begin to bulge out, and water will begin to diffuse out of the cell wall, out of the membrane. Eventually what will happen is that you will have significant amount of water that diffuses out, and the membrane will rupture and the cell will lyse. And this is basically going to destroy the cell. If we look over here at the photographs, these are healthy cells, healthy, generally gram-positive organisms that are untreated. Once they're treated and you go through cell lysis, you still have some cells that are intact, but here all of this kind of spongy, stringy looking stuff are cell residues that show a number of cells that are undergoing lysis and cell death. So these are obviously susceptible to the effects of penicillin. You have some antimicrobials that will also inhibit nucleic acid synthesis. Now remember that nucleic acids are going to be RNA, DNA, and ATP. So we can have any of those being inhibited with, and they will interfere with nucleic acid synthesis. They can block the synthesis of nucleotides. That means that those nucleotides don't get built. And if there are no nucleotides that are being built, then you have no way to make your nucleic acid. It can inhibit replication, whether that replication is the replication of DNA or whether it is the replication of messenger RNA. So you can target DNA replication as well as transcription. It can stop transcription, and when it stops transcription, what it does is it interferes with the ability to produce messenger RNA. And then when we go into inhibition of DNA synthesis, we lapse over into interfering with cell division, because if we can't synthesize our DNA, we can't go through cell division. We also have some drugs that will block protein synthesis. Basically, they are going to inhibit translation by interacting with the ribosome and the mRNA at some point. One of the nice things about protein synthesis inhibitors is there is a difference between the prokaryotic and eukaryotic ribosome. Therefore, our targets are different. And so these protein synthesis inhibitors can be very good at selective toxicity, because if their target is the 50S or the 30S ribosome, well, as a eukaryotic organism, we don't have a 50 or 30S ribosome, so we don't have a target, and therefore our protein synthesis is not interfered with. Here are 4 different, your book actually shows 5, and I couldn't fit all 5 on this page, so I took the 4 most common. Here are 4 different protein synthesis inhibitors. You've got the aminoglycosides, and basically what they do is they insert themselves into the 30S ribosome and basically cause misreading of the messenger RNA, and you have an incorrect protein that is non-functional, and the cell is going to die. Chloramphenicol, basically what it does is, you'll see here, it binds to the 50S ribosome and it blocks the bond that you will get made between the tRNA in the P site and the tRNA in the A site. No peptide bond will be formed, and therefore no protein can be produced. Tetracyclines, they are going to bind to the 30S ribosome. They are going to block the tRNA so that the tRNA can't come into the acceptor site or the A site of the 50S ribosome, and because the tRNA can't get in, there is no amino acid going to be brought in, and the mRNA can't be read. And then finally, erythromycin also binds to the 30S ribosome. It is going to prevent the ribosome from moving down the messenger RNA. This is kind of like putting a stub or a block in the way of a tire. This cell isn't going to be able to move down because it can't access the next messenger RNA, and it blocks and prevents translocation of the messenger RNA. You do have some cell membrane disruptors. Basically what they are going to do is damage the membrane, which will invariably cause the lysis or disruption of that membrane, and things will fall apart, and the extracellular fluid flows in, the intracellular fluid flows out, the cell lyses and dies. There is some specificity for particular microbial groups based on the differences in the lipids in their cell membrane. So not all cell membrane inhibitors will work equally on every cell. So they are going to rely on knowing the structure and the specificity of the lipids in the membrane. Folic acid synthesis inhibitors. These are where we need to go back and remember our competitive inhibition from the metabolism section. These are the metabolism inhibitors. Sulfonamides and trimethaprons, they act as competitive inhibitors for the substrate for a particular enzyme that converts the substrate of para-aminobenzoic acid, or PABA, to folic acid. And if we look here, on the diagram, you'll see that PABA is going to be converted through a system of enzymes that essentially will end up turning it into folic acid, which is basically a coenzyme for the synthesis of the purines, the pyrimidines, and a number of amino acids. So if we can break the cycle along this pathway, we can shut down the process of folic acid production, and the bacteria will die. This is another one of those cells that has a good, pardon me, a good selective toxicity, because we get our folic acid from from the foods that we eat. Bacteria make their own. And so by adding something like a sulfonamide drug, which is called a metabolic analog, we can control the amount of folic acid that gets produced within the cell. When you have an enzyme that is open, that substrate's going to bind in there, and you're going to end up with your folic acid product. If we have a sulfa drug like a sulfonamide or trimethoprim, these are going to inhibit this folic acid at some point here on the pathway. If we use a sulfa drug, we're going to basically stop it at the first pathway, because what it does is it inhibits the binding of the substrate completely. This sulfa drug binds into the enzyme, and the substrate can't bind, so you get no product. You're going to have a constant flow of the sulfa drug so that it can continually occupy that substrate for the treatment, so that folic acid doesn't get made, and you begin to decrease the number of bacteria that you have in your system. Now, trimethoprim actually is going to interfere not at the substrate level where PABA does, but rather here at where the dihydrofolic acid is created. Dihydrofolic acid is a product of this conversion of PABA, and what the trimethoprim does is it is a competitive inhibitor of the enzyme dihydrofolate reductase. If we can stop that, we stop the production of folic acid, and again we stop the production of our purines, pyrimidines, and amino acids to our bacteria. Here is a little bit bigger picture if you want to see it. Only this first portion is done by bacteria. Trimethoprins, we do have a little less selective toxicity, because we do have this whole system of dihydrofolate reductase, where we turn the dihydrofolic acid into folic acid, so that we have folic acid, but we get it right. This dihydrofolic acid we get from our diet. But anyway, here's a bigger picture, because it's not all that great on that previous slide. If we look at the antimicrobial drug groups, we have about 260 different antimicrobials. They're going to be classified in about 20 different drug families. We're not going to go through all of the drug families, but there are a lot of drugs out there. And a good number of them are ineffectual because of antibiotic resistance or some other type of microbial interference. So we're going to talk a little bit about that towards the end of this lecture. The largest number of antimicrobial drugs are for bacterial infections. And of course, that's because we come in contact with bacteria more often than we come in contact with some of these other organisms. They also are going to be those that are going to give us the widest range of infectious agents. Most of us come in contact with fungi, molds, things like that, and they don't really cause us a lot of trouble, a lot of harm. But bacteria tend to do that. So most of our antimicrobials are going to be for bacteria. If you look at this this table here, you'll see this is basically the spectrum of activity of antibiotics. You've got a number of different diseases here. Some of them are from the mycobacteria group, tuberculosis, things like that. Gram-negative bacteria, gram-positive bacteria, chlamydias, rickettsias. These are all going to be different types or groups of bacteria. And this is the spectrum of activity of a lot of these different drugs. If you look here, isoniazid doesn't really do a whole lot for anything but the bacteria in the mycobacteria family. Streptomycin takes care of the mycobacteria as well as the gram-negatives, a good number of them. Tropomycin, again, does a little bit with the mycobacteria, a little bit with the gram-negative bacteria. Polymyxin, purely gram-negative. It's only going to work with these gram-negative organisms. We're going to talk a little bit about bacitracin, which is actually one of the same groups of drugs as polymyxin. Carbapenemines, these are going to be those drugs that are pretty wide spectrum, pretty broad spectrum. You'll notice that all of these up here, isoniazid, narrow spectrum, streptomycin, relatively broad spectrum, tropamycin, relatively broad spectrum, polymyxin, narrow spectrum. These carbapenemines are basically very narrow, very broad spectrum. They're going to cover both gram-positive and gram-negative. If we look at the tetracyclines, we're going to see these tetracyclines cover a whole multitude of sins here. These are what we would call a very broad spectrum antibiotic. The sulfonamides, the cephalosporins, the penicillins, again, primarily going to be with these gram-positive organisms, but they're going to take care of the gram-negatives somewhat as well. But you can see here, are there normal biota in this group? Yes, we have some bacteria that are part of the normal flora in all of these groups, except for this last one with the rickettsias. This is a much better picture. This picture is in your book on page 336. If you'll just kind of follow it drug by drug, you can kind of see broad spectrum versus narrow spectrum, and do they interfere with some of these normal flora as well. So this is a good graph. Let's talk about some very specific drugs now. We've kind of talked about all the targets and what those targets are, but let's talk about some of the drugs that are in those targets. When we're talking about antibacterial drugs that target the cell wall, the penicillin group is probably the most significant. Most of these drugs are going to end in the suffix cillin or cillin. They can be obtained through a natural process, through microbial fermentation, through the fungi. They are going to consist of three parts. They have a thiazolidine ring, which is basically this 5-ring structure, the yellow one here. Then they have a beta-lactam ring, and then they have a variable side chain. And the side chain is what's going to make each penicillin. These are basically the R group, if you will, of each of these penicillins. The thing that is most important is this beta-lactam ring here. A good number of our bacteria that have become penicillin resistant is because they have the ability to break this beta-lactam ring. And when they break it, let me see if I can get my cursor back here. Here you go. When they break this ring, it essentially cuts the molecule in half, and now it becomes completely non-functional. Here are a number of different penicillins. You've got penicillin G, penicillin V, oxacillin, methicillin, ampicillin, amoxicillin, carbenicillin, and a number of different other ones here. Azocillin, I guess, and mesocillin. You'll see that they are divided into narrow spectrum versus broad spectrum. What they work on, you'll see that these narrow spectrums are going to be primarily against the gram-positives. The broad spectrums here are going to work against gram-positives and gram-negatives, and then the very broad are going to be working on some of these other organisms like these pseudomonads that are that tend to be very penicillin resistant. The cephalosporins are a newer group. Well, I shouldn't say they're a newer group. They actually were isolated in the 1940s from a mold called Cephalosporium, and I can never pronounce this name, acremonium, I believe is the way it says. And all of these cephalosporin drugs contain a beta-lactam ring. They do account for the majority of all of the antibiotics that are administered now. Fewer people are allergic to cephalosporins than they are to penicillins, and so a lot of these drugs are going to be the cephalosporins. One of the characteristics about the cephalosporin is they do have this basic beta-lactam ring. They've also got this 6-sided ring here, but they have 2 R groups now, and so you have this same beta-lactam nucleus with different R groups, and these are going to give you the different generations, if you will, of this drug family. All of these generations are essentially going to denote a specific type of reaction that, or a type of drug that that these organisms will work against. So just like you have with the penicillins, you have first, second, third generation penicillins. You also have these that are going to be based on their antimicrobial or antibacterial activity. What do they tackle the best? And so your first generation may be very good at killing a particular group of organisms, where your fifth generation may look at something much more much different. You do have some subgroups of cephalosporins, some subgroups and some uses. These are very broad spectrum, and a lot of them, and this actually should say most and not mot, they mean, when they're broad spectrum, meaning that they are going to kill more than just gram positives or just gram negatives. They are resistant to most penicillinases, so that penicillin or that beta-lactam ring is going to stay intact, and those cells then are going to be able to survive an attack by these bacteria that may be producing these penicillinase enzymes. Cephalosporins cause fewer allergies than penicillins, and the generation, there are 5 generations, not 4. There are 5 generations of antimicrobials that that are very effective against a wide variety of organisms. You do have some other drugs that target the cell wall that are not necessarily in the penicillin group. They don't have that beta-lactam ring. The first one is bacitracin. This is produced by the bacterium Bacillus subtilis. We've used this in the lab a number of times. Basically, it is good for skin infections, superficial skin infections. It's used in your triple antibiotic, oftentimes combined with neomycin and polymyxin. These are very good at targeting the cell wall. Isoniazid, this is used for the treatment of active tuberculosis. A lot of times they'll take isoniazid, combine it with 2 or 3 other drugs, and this is going to help with the prevention of resistance for these drugs. When we get into the drug portion of the class, or excuse me, the disease portion of the class, we're going to see that treatment for active tuberculosis is prolonged. It can be 6 months, 12 months. And so when you are exposed to a drug that often, you can often become resistant. The organism can become resistant. So by combining this, we can prevent that resistance. Vancomycin is kind of been called the last drug of choice. If everything else has failed, they're going to go to vancomycin. It is a very narrow spectrum antibiotic. It targets the gram-positive cells only, particularly those in the staphylococcal family. It's effective at treating staphylococcal infections that are penicillin and methicillin resistant. So if you know somebody that has a MRSA that is not being treated, or is not other drug cocktails are not killing it, they're going to place this person on vancomycin. The interesting thing about vancomycin is it's a very good drug, very toxic drug. I had it when I was in the hospital after my brain surgery, and it's a very kind of a painful drug to take, because it does burn the vasculature as it goes through an IV. You have to take it through IV, because vancomycin, if swallowed, getting into the stomach, the acid of the stomach will break the vancomycin, and it is no longer effective. Fosfomycin, this is basically an alternate treatment for urinary tract infections that are caused by enteric bacteria like E. coli. These organisms are often caused by just fecal matter getting into the urinary tract, and these fosfomycin drugs are very good at killing those organisms. Drugs that are going to target protein synthesis have a number of different ways that they do that. You saw on that graphic that I showed you a few slides earlier, there were 4 different drugs, and they target things differently. They target it at the 50S ribosome, they target the 30S ribosome, they target the tRNA or the mRNA. So there are lots of of ways that these drugs can inhibit protein synthesis. The first drugs are the aminoglycosides. These basically are made from a species of cells that are found in the soil called actinomycetes. And the genera that are most actively are most active in the production of these aminoglycosides are the genus Streptomyces and Micromonospora. These are going to be the two that give us the greatest effective aminoglycosides. They are relatively broad spectrum because they do inhibit protein synthesis. If they have a cell that is gram positive, gram negative, protein synthesis stays the same in each type of cell. So because they do target protein synthesis, they are relatively broad spectrum. There are some subgroups and uses. Aerobic gram-negative rods and certain gram-positive bacteria are going to be susceptible. Streptomycin is very good. Streptomycin is a protein synthesis aminoglycoside drug. Is very effective at controlling bubonic plague and tularemia. It is also a good anti-tuberculosis agent. This is one of those drugs that they will combine with isoniazid to treat active tuberculosis. You know, we don't think about bubonic plague, but we actually do have small outbreaks of bubonic plague in a number of different places throughout the world, and this is a good way to effectively control it. Gentamicin is less toxic than streptomycin and some of the other aminoglycosides, and this is a little more narrow spectrum than some of the others, and it's used primarily against gram-negative rods. And then neomycin, this is one of those triple antibiotic drugs. Bacitracin is a cell wall synthesis inhibitor. Neomycin is a protein synthesis inhibitor, and we're going to see that polymyxin is a cell membrane disruptor. So you kind of target a lot of things with that triple antibiotic. Tetracycline is probably one of the most effective drugs. We saw on that graph that it is a very broad spectrum antibiotic. It binds to the ribosomes and blocks protein synthesis. Extremely broad spectrum. Its subgroups and uses are going to include gram-positive as well as gram-negative rods and cocci. Aerobic versus anaerobic bacteria, it doesn't matter. It can also be used to treat some of these archaebacteria like Mycoplasma, Rickettsia, spores, and some of the spirochetes that are very pathogenic and are part of the sexually transmitted disease groups. Doxycycline and minocycline are used for those sexually transmitted diseases. Rocky Mountain spotted fever, Lyme disease, typhus, of all things. You don't ever think about that, but again, in certain parts of the world, typhus is a huge problem. Pneumonia caused by mycoplasma, cholera, leptospirosis, acne, and even some protozoal infections are going to be controlled with tetracycline antibiotics. Doxycycline and minocycline are two of the primary tetracyclines. Doxycycline is actually the drug of choice for anthrax infection. They found the CDC actually kind of changed their recommendation from ciprofloxacin to doxycycline because doxycycline is so much more broad spectrum than ciprofloxacin. Chloramphenicol is another one of the protein synthesis inhibitors. It has a very unique structure. It's what they call a nitrobenzene. These can be very toxic to human cells because of the benzene, and so it's not going to be used very broadly. It's going to be used only in a very restricted sense. Basically what it does is it blocks peptide formation. It binds to that 50S ribosome, and it blocks those peptides from that peptide bond from being formed between the two amino acids during protein synthesis. This is an entirely synthesized antibiotic. There is nothing natural about it. It is a chemical process. Benzene is certainly not a naturally occurring molecule, so this is going to be one of those that is made in the laboratory. Erythromycin and clindamycin, these are two pretty common bacteria. Erythromycin has what they call a lactone ring rather than a beta-lactam ring. It is relatively broad spectrum. It does have a relatively low toxicity, so you can give it without a whole lot of worry that it's going to cause a lot of problems. Basically what it does is blocks protein synthesis by binding particularly to the 50S ribosome. It's going to be used in like the Mycoplasma pneumonia, Legionellosis, this is the Legionnaire's disease, chlamydia infections, those are sexually transmitted disease, pertussis, and diphtheria. So some of these kind of uncommon diseases are very effectively controlled with erythromycin. Clindamycin, erythromycin is one of the things that they use erythromycin for, as well as tetracycline, is acne. And it's very effective with that. Clindamycin is broad spectrum. It does cause some adverse reactions in the gastrointestinal tract, particularly colitis, diarrhea that gets to the point of colitis. So there is going to be some restricted applications for this, and it's not going to be one that's used quite as widely as maybe erythromycin or one of the other tetracyclines. Cindercid and oxazolidones, these are also protein synthesis inhibitors. These are going to be effective against Staphylococcus, Enterococcus, and some very resistant strains of Streptococcus, like some of the streps that can cause strep throat or necrotizing fasciitis. What it does do is it binds to the 50S ribosome, and then it inhibits translation from that point. If it's binding to that 50S ribosome, it is inhibiting the ability of the ribosome to read the mRNA, and translation stops. Oxazolidones, these are going to inhibit the initiation of protein synthesis, so it doesn't even begin. It's not found in nature. It is a drug that is synthesized in the lab. It is one that there is not a whole lot of drug resistance to it now, so you don't have a lot of bacteria that are resistant, and hopefully with the use with responsible use of this, resistance will be slow to develop. It usually will develop, but hopefully at a very slow rate. This is going to be used to treat infections that are caused by some very significant clinical pathogens. Number one, MRSAs, which are methicillin-resistant Staph aureus, and then vancomycin-resistant Enterococcus, or VREs. These are both organisms that will usually be treated with something like vancomycin. But in order to kind of keep vancomycin from becoming drug resistant, or microbially, those microbes becoming drug resistant, some of these oxazolidones, these are going to be the ones that are going to kind of take the place of vancomycin and start fighting some of these staphylococcal and enterococcal infections. We do have some drugs that will target metabolic processes like the production of folic acid or folic acid synthesis, and these are going to be the sulfonamides, the trimethoprim, and the sulfones. Not cell phones, sulfones. Sulfonamides, these are what we call the sulfa drugs, and this is the first modern antimicrobial. It's the first one that we knew the mode of action for. We knew that it was a competitive inhibitor of that para-aminobenzoic acid. It is a synthetic drug. It is not one that is found in nature, so it's one that we can use and make within the laboratory. It's used for shigellosis or acute urinary tract infections, particularly those that are caused by enteric bacteria. It also is very effective against certain protozoal infections, so it does kind of jump the fence from bacterial to protozoal. Trimethoprim, these are going to inhibit the step that will initiate that final step of folic acid synthesis. So it inhibits any of these drugs that get past the sulfonamides in the synthesis of folic acid. It's going to stop it at the very last step. This is going to be often given in combination with sulfamethoxazole. A lot of times, if you look at your prescription of sulfamethoxazole trimethoprim, it'll say XS times T. And this basically kind of is giving a 1-2 punch to the folic acid, the production of folic acid. If we don't stop it at the very start, we can stop it at the end, and it will keep folic acid from being produced. One of the primary treatments is for pneumocystis carinii. This is basically pneumocystis pneumonia in AIDS patients. The sulfones, these are chemically related to the sulfonamides. They aren't nearly as broad spectrum as the sulfa drugs, but they are very good at treating the disease, Hansen's disease, which is our modern day leprosy. Antimicrobial drugs that are going to target DNA and RNA are primarily going to be those drugs called the fluoroquinolones or ciprofloxacin. There are a number of different drugs that are going to be part of this fluoroquinolone family, but I just want to talk about ciprofloxacin in particular, so that we're not bogged down in all of these different drugs. Fluoroquinolones are high potency. They are broad spectrum. They are going to inhibit a wide variety from gram-positive, gram-negative, even some of these other species that may have a bit of a different cell wall structure. You don't have to have a very high concentration of fluoroquinolones for them to be effective. Side effects can be rather dicey. They can include seizures and other brain disturbances. So they aren't this drug isn't given randomly without thought to the particular patient. There are some. The ciprofloxacin is a second generation quinolone. It's used primarily to treat urinary tract infections, STDs, gastrointestinal infections, osteomyelitis, respiratory infections, and soft tissue infections. Cipro probably made its greatest appearance on the national stage when it was used to treat the anthrax scare when we had the in 2001 when the anthrax was mailed through the post office. People that were potentially exposed were prophylactically treated with ciprofloxacin. It was just shortly after that, though, that the CDC actually changed their recommendation from ciprofloxacin to doxycycline, and it's only because the tetracycline drug, doxycycline, much broader spectrum, and they also wanted to try and limit the potential for ciprofloxacin resistant bacteria. Rifampin is another one of the DNA RNA drugs. It's produced by the genus Streptomyces. It is a limited spectrum antibiotic. It's going to be used mainly in the affection of gram-positive rods and cocci and a few gram-negative. It's a mycobactericidal drug. It's going to be effective in the treatment of tuberculosis and leprosy, and it's usually going to be in combination with a number of different drugs like doxycycline, streptomycin, any of those others. One of the side effects that's worth noting here about rifampin is that when you consume rifampin, when you take rifampin, it actually binds to the mucous membranes, and all of your bodily fluids actually turn orange. So you sweat orange, you pee orange, you cry orange, you blow your nose and it's orange, your saliva is orange. So it's got some pretty significant side effects. That's probably a great parlor trick for the first couple of days that you take it. But if you're on rifampin for tuberculosis infection, you're on it for about 6 months, and I'm sure after 6 months that orange mucus gets pretty old. Drugs that target the cell membrane are going to include things like polymyxin and daptomycin. Polymyxin is a narrow spectrum antibiotic. It's from the bacteria Bacillus polymyxa. Limited usage due to the fact that it is very toxic to the kidneys. The kidneys are, if you think about the tissue of the kidneys, lots of smooth endoplasmic reticulum. That smooth endoplasmic reticulum is a lipid. These polymyxins are going to bind to the lipids of the membranes and they're going to disrupt them. Polymyxin B and polymyxin E are used to treat drug-resistant Pseudomonas aeruginosa. So some of these polymyxins can be used topically. Some of them can be used through an oral ingestion. Daptomycin, this is basically going to be made also from the bacterial genus Streptomyces. It is most active against the gram-negative bacteria, primarily because of that outer lipid membrane Remember, those gram-negative cells have that lipid membrane that is on the outside. It's going to bind to that and begin to destroy that cell membrane and work its way into the plasma membrane. So that's all of the antibiotics. One of the things that I would encourage you to do is to take some time and go drug by drug and put all the pertinent information of each of those drugs onto a card. I'm not a great fan of flashcards, but I do think this is material that lends itself to flashcards. This is going to be that kind of memorization type stuff, and hopefully those of you that have been struggling and need some memorization type activities, this is for you. Let's talk a little bit about fungal infections. Fungal cells are eukaryotic, so selective toxicity can be a problem. The majority of chemotherapeutic drugs are designed to act on bacteria and therefore are ineffective for fungal infections. There are a number of similarities between fungal cells and human cells, and this makes these agents a little more toxic to humans. Amphotericin is one of a very common antifungal drug that the side effects of it are so bad. The name that has kind of been dubbed by the medical community is amphoterrible. So you can see that there are a number of different complications that you can have. There are four main groups of the antifungals. You've got the macrolides, you've got griseofulvin, the synthetic azoles, and flucistonacin, I think that's how you say that, flucistonacin. I'm not going to go into a lot of the antifungals just simply because there are so many, and I think again we need to direct our time to the antimicrobials, the antibacterials, I mean. Antiparasitic chemotherapy, you basically have drugs like the anti-malarial drugs, quinine and the relatives of quinine. Quinine is actually extracted from the bark of a cinchona tree. This is a tree that is in the areas that are endemic to malaria in Asia and parts of the Philippines, things like that. Basically, they have replaced it by synthesizing these quinolones, so chloroquine and primaquine. These are less toxic to humans. Quinine can actually kill you if you are dosed with it incorrectly. So these synthesized quinolones actually are a better antiparasitic drug than the original quinine. You have some chemotherapy for other protozoans other than malaria. These are going to be amoebacidic. These are going to be ones that are going to kill some of these amoebas. Metronidot. Oh gosh, you guys are. This is killing me. Metronidazole. This is also called Flagyl. A lot of times when people have gastrointestinal diseases or problems, a lot of times they will give them Flagyl to try and kill any sort of parasitic infection that they may have. This is also going to be used for treating mild to severe infections with Entamoeba histolytica. This is one of those, it's a liver fluke. It can be taken orally and also used to treat infections with Giardia lamblia and Trichomonas vaginalis. So these are going to be good drugs for those parasitic infections. Quinacrine, sulfonamides, and tetracyclines are also good for antiparasitic therapy. So not all of these drugs are going to be purely antiparasitic. Some of them are actually antibacterial. Antihelminthic drug therapy, these are going to go towards the treatment of flukes, tapeworms, roundworms. These are going to be the ones that have the greater physiology to the human and therefore, can be a little more tricky because of this issue of selective toxicity. Most of these are going to use, most of these drugs are going to block reproduction of the helminth in some sort. It's not very successful, though, in eradicating the adult worms. it's not a good process. You're actually going to try and immobilize or inhibit the metabolism stages within the stages of the life cycle. The thing about helminths is they have different life cycles in different bodies, even in different organs within the body. So sometimes it's hard to target those. And so by blocking reproduction, you don't always eradicate the adults. You may eradicate some of the other parts of the life cycle, but the most effective way is to immobilize or disintegrate or inhibit metabolism at some portion in the life cycle. Antiviral chemotherapeutic agents it is very difficult for selective toxicity to be achieved because the metabolic system for the virus and the metabolic system for the host are the same system. So if you kill the metabolic system of the virus to kill the virus, you're killing the host. There are several antiviral drugs that have been developed, and they target several points in the infectious cycle. Amantadine, this is going to inhibit virus entry. If it can't enter, it can't replicate. Acyclovir, very common drug, inhibits inhibition of nucleic acid synthesis. Whether the virus is an RNA virus or a DNA virus, it doesn't matter. It's going to interfere with that nucleic acid synthesis. It can't have replication of those early proteins, those late proteins, and the cell is going to die. Indinavir, this is one of the drugs that is used in HIV treatment. This is going to be inhibition of viral assembly and release. You may be able to make the parts, but you can't put the parts together, and if you can't put the parts together, the virus can't be released, can't be sent out to infect new cells. There are three major modes of action. You're going to bar penetration like you have with amantadine. You're going to block transcription translation of these viral molecules, which is going to be inhibition of nucleic acid synthesis and even protein synthesis, and then prevention of maturation of these viral particles like you do with indinavir. You do have an alternative to some of these artificial drugs. There is the glycoprotein interferon. This is basically made from the fibroblasts and leukocytes in the blood. Basically, it's made in response to a various number of things, particularly viral infections. We can make interferon through a process of recombinant DNA technology. We can take interferon from one organism and we can put it into another organism and make a lot of interferon. Number of therapeutic benefits: it reduces the time of healing and some of the complications that you have with certain infections. It's known to help prevent and reduce some of the symptoms of cold and papillomaviruses. It also will help in the slowing process of certain cancers. You can turn these, especially if these cancers are caused by an antimicrobial, you can actually slow down the metabolism of this whole process. It also is used in treating a rare cancer called hairy cell leukemia, which is a very rapidly growing leukemia, hepatitis C, genital warts, Kaposi's sarcoma in AIDS patients, and a number of different other types of severe infection like that. There are some very serious side effects. Sometimes the side effects are not as beneficial as the drug itself, and so people a lot of times will choose to go off of interferon treatment. Lots of times there will be personality changes, and generally not for the better. Lots of anger management issues, lots of psychosis. You can also have dysfunction of the immune system. The immune system doesn't recognize that it needs to start picking up the ball and will kind of sit back and let this interferon do what it needs to do, and it will become overwhelmed and the immune system doesn't pick up and help out. So that's essentially all of the drugs that we're going to talk about. I know that seems like a lot, but I think once you start breaking it down drug by drug, you're going to see it's not too many. What I want to talk about now is drug resistance. Basically, if we try and destroy a bacteria, that bacteria is going to try and figure out a way to circumvent the action of any drug that's in its presence. So drug resistance is essentially an adaptive response in which a microorganism begins to tolerate the amount of a drug that would ordinarily be inhibitory. This can be intrinsic, meaning that it's something natural to the bacteria, or it can be acquired either through mutation or transformation, transduction, conjugation, any of those things. You do have some specific mechanisms of drug resistance, and I'm going to look at some of those here. If we look at drug inactivation, basically you have a bacteria that produces an enzyme like a penicillinase. You have an active penicillin here. The bacteria produces penicillinase. It breaks that beta-lactam ring, and now we have an inactive drug. We can have decreased permeability. Basically, you have the drug that binds to specific receptors on the cell, and what happens is through mutation or alteration of some sort, those receptors change, and now the drug doesn't bind to any of them, and the cells will survive. You have the activation of pumps. There is a particular drug-resistant strain of bacteria to tetracycline that produces this drug pump. You have on its surface, you have receptors for those drugs to bind to the cell. They get into the cell, and what the cell will do is it will begin to produce a pump. This microbe will get into the cell, and then it's pumped almost directly straight out through this cell pump. You can also have changes to the drug binding site. This is particularly one of those resistance mechanisms used in protein synthesis inhibition. Basically, the binding site of the target is altered so the drug doesn't have any effect. If you look here, you'll see that we have our little triangle here, but if the ribosome, in this instance, which is the target, changes that binding site on the target, it now doesn't fit the drug, and the cell can continue on that whole protein synthesis mechanism. It can also use an alternate pathway. This is the way some of these sulfa drugs can end up being microbes being resistant to the sulfa drugs, is if the pathway is interfered with, it can actually develop a new pathway, and the outcome will be the same. You'll get the product that you need, and the cell will continue to live on. I love this picture. This picture is actually in your book on page 347. And it gives some very good explanation of what's going on with each of these different mechanism of resistance. When we talk about the natural selection of drug resistance, basically what we're saying is that you've got, within a population of bacteria, you have members that harbor a mutation that are going to confer drug resistance. So if we look at our picture here, all of these dark cells are non-drug resistant, but we just have a couple that have that drug resistant mutation. With some sort of environmental pressure, you're going to inhibit the growth of all of the non-resistant strains, and only the resistant ones will grow. So if we come over here and we expose these cells to the drug, which is going to be that environmental or selective pressure, we're going to see that our sensitive strains are dying, and our resistant strains are multiplying. If we continue to allow that population to grow, eventually the mutated ones will outnumber the non-resistant strains, and they will become the dominant strain in the environment, and you will then now have a nearly fully drug-resistant strain of bacteria. There are some other approaches to antimicrobial therapy that are not quite so chemically based. A lot of researchers are trying to find new targets in which the bacterial cell can be destroyed. Things like targeting iron scavenging capabilities of the bacteria. Bacteria are going to consume iron from their host. If we can stop that, then that's not going to be something that is quite so changeable within the organism. It's not going to have quite the effect on the host that some of these other microbial drugs have. Targeting some genetic control mechanism what is called a ribo switch that can turn protein synthesis on and turn it off. Some other things that they're looking at are probiotics, help to reestablish normal flora so that you can get rid of those other organisms and reestablish that normal flora after broad spectrum therapy so that you're not going to be down quickly. That's part of the problem with some of these, is that they are going to basically destroy all of the flora, and you are not going to have the protection that you need. So having some of these probiotics to help it reestablish normal flora. One of the things that a lot of doctors are doing with women that are on therapy for broad spectrum therapy for vaginal yeast infection is to eat yogurt while they are on this therapy so they can reestablish that flora. Prebiotics, these are going to include certain nutrients in your body. These are going to help to encourage those beneficial bacteria. These particular bacteria, they're called, or these prebiotics are called fructans. There's a commercial out, and I can't remember the name of the drug or the prebiotic, but it actually is one that is going to help to reestablish or to begin to establish a better bacterial flora so that you are not at quite such a loss. Lantibiotics, these are short peptides that are going to be produced by bacteria that are going to inhibit the growth of other bacteria. So if we can put good bacteria that have these lantibiotic properties, they can shut out the bad bacteria and basically keep you protected. Let's talk a little bit about the interaction of the drug and the host. One of the characteristics of a lot of antimicrobials is that they have some sort of adverse interaction with the host. 5% of all people taking antimicrobials are going to experience some adverse reaction. There are three major areas of interaction. These drugs are going to be toxic to organs, they're going to cause an allergic response, or they're going to cause the suppression or alteration of your bacterial flora. Let's look at toxicity to organs. This is probably the one area where most of the damage is done. You can have some very adverse effects to a number of different organs. Talking about the liver is responsible for metabolizing these drugs and detoxifying the blood. If we have injury to the liver, this can result in hepatitis and liver failure. The kidney, this is responsible for filtering and excreting the drugs. These drugs can also interfere with filtration. Some of these drugs can actually crystallize in the tubules and will form stones, these kidney stones. And if you've ever had kidney stones, you know how painful that is. This often will obstruct the flow of urine, so you end up also beginning to initiate kidney infections, urinary tract infections. Gastrointestinal tract, the most common complaint is going to be some sort of diarrhea. If this diarrhea continues to progress, it can actually progress to colitis. The most usual complaints, though, are going to be caused by the disruption of some normal flora in the intestinal tract. The nervous system, you can have some drugs that directly affect the brain, and these are going to result in seizures. You can also have nerve damage, generally to the 8th cranial nerve, and this is going to lead to dizziness, deafness, as well as some motor and sensory disruptions. If you can remember what the 8th cranial nerve is, you'll understand why dizziness and deafness are two of the main complaints. You can have toxicity to the skin. Basically, this is usually a symptom of a drug allergy or some sort of direct toxic effect. So the skin is certainly not something that you should ignore when it comes to looking at the effect of bacteria on your organs. The teeth, it doesn't seem that the teeth would be very impervious, but they are. Tetracyclines in particular are going to bind to that tooth enamel, and they're going to cause some gray or brownish discolorization in the teeth. The prescribing of tetracycline is contraindicated in children under the age of 8 for the simple fact that their teeth are still soft, they're beginning to develop, and they are more susceptible to this discolorization. Other systems that are affected are going to include the cardiovascular system, blood-forming tissues, respiratory tract, as well as the bones. If you look here, this is a picture from your book on page 353. You'll see this kind of brown or gray band across the teeth, and this is essentially from it binding to the enamel in the teeth, and this is permanent. You can't brush it out, you can't polish it out. Actually, this guy's teeth actually look pretty good here. The gums look a little receded, but other than that, they look pretty good, and you can't do anything to get rid of it. The second interaction are going to be allergic responses to drugs. The most frequent drug reaction is going to be an allergy. A reaction occurs because a drug acts as an antigen. When we start talking about the immune system, we're going to recognize that an antigen is a molecule that is going to stimulate a response by the nervous system. Generally, antigens are something that are foreign to the body. The body doesn't like it, and it's going to react. Allergic reactions have been reported for every major type of antimicrobial. So there isn't an antimicrobial out there that someone isn't allergic to. Penicillin accounts for the greatest number, followed by cephalosporins, or excuse me, sulfa drugs, sulfonamides. The body, essentially in an allergic reaction, becomes sensitized on the first exposure. Oftentimes there are no symptoms with that first exposure. It's with the second exposure that you begin to see a reaction, and reactions can go from a minor skin irritation to respiratory inflammation and difficulty breathing to full-blown anaphylaxis, which is essentially an allergic response that develops extremely rapidly and can lead to death. You end up with multiple organ failure, inability to breathe, respiratory inflammation. It's horrible. The third group that we want to talk about is the suppression and alteration of the normal flora. When we talk about biota, this is basically the normal or resident flora that we have on and in the body. It is usually harmless, and it is beneficial most often. It is a small number of them can be pathogenic, particularly if they get in an area where they shouldn't be to begin with. If a broad-spectrum antimicrobial is used, it is generally going to destroy both the infectious agent as well as this normal microbiota. When you have some of these beneficial species are destroyed, you have microbes that were once kept in a very small number begin to overgrow in another area. And this is what is called a superinfection. And basically, using a broad-spectrum antibiotic for a urinary tract infection destroys the bacteria called lactobacilli of the vaginal tract. And without this lactobacilli, these Candida albicans can proliferate. They can grow out of control, and they cause a yeast infection. You can also have the same thing happening in the mouth with thrush. You can end up with thrush. Oral therapy with a good number of tetracyclines and clindamycin are associated with antibiotic-associated colitis. There is a picture, this picture actually right here in your book, shows what happens when you have an infection in the throat or something, and you're given a broad-spectrum antibiotic. That broad-spectrum antibiotic also kills the organism that's causing the throat pain, but it kills everything else throughout the body as well. So it's going to go in and it's going to destroy the beneficial biota, and it's going to leave those potential pathogens to grow, and you can end up then with the overgrowth of a pathogen in another part of the body, and this is called a superinfection. There are some factors or some considerations that we need to take when we're talking about antimicrobial drugs. 3 factors have to be known. What is the nature of the organism? Is it a gram-positive? Is it gram-negative? What kind of organism is it? The degree of the microorganism's susceptibility to the drug. If my microorganism is resistant to the drug, then I'm in big trouble. And then the overall medical condition of the patient. Some patients are in such a state that using a very aggressive antimicrobial could be detrimental. And then identifying the agent. direct examination of body fluids, sputum, or stool samples will allow for a rapid initial method. A lot of times when you go to the doctor and they start looking at the back of your throat, say you think you have strep throat, they see those pus pockets at the back of the throat. That is direct examination, and they can begin therapy right away, and they can do some of the testing a little bit later to determine, yes, you do have strep throat, or no, you don't. We need to change your drug. The choice of drug is going to be based on the experience with the drug. Have you had an allergic reaction before? Has it been effective against this particular microbe? This is basically what they call the informed best guess. So we need to be able to see, this is what's worked before, let's try it again. We also need to have some testing for drug susceptibility. You've done some of this testing in lab. Here are two tests that are used for drug susceptibility. The Kirby-Bauer Distaffusion test, where you have your Mueller-Hinton agar, you make a smear all over the plate to grow a lawn of growth. You put your discs of your particular antibiotics on there, and you then let it incubate, and then you will measure these zones of inhibition. The larger the zone of inhibition, the more effective the drug. But just because it has a large zone does not necessarily mean that it is going to be sensitive. There is a standard that is measured, and you will see here under each one of these you have R less than a certain number, S is greater than a certain number. And that basically means that if it's smaller than this, it's resistant. If it's this or greater, it's sensitive. And so when you measure those, based on this standard, you will be able to determine whether or not this drug is good for your particular organism. If you see here, Norafloxacin is one of those ciprofloxacin, those fluoroquinolones. You'll see that it has a zone of inhibition of 27 millimeters. So it is larger than the sensitivity millimeter range. So this is sensitive. You'll see this one says I and this one says I. That means that they are intermediate. That means it's between 17 and 21. So if it's between 17 and 21, these are going to have an intermediate resistance. They are not the best drugs to use, but in a pinch, you could use them. Any of these that have an R on them, they are resistant. If you look here, you'll see that this zone of inhibition is bigger than this zone of inhibition. This one right here that is on that has the C30, the chloramphenicol 30. It is fully resistant to whatever this organism is. The ampicillin is intermediate right here, and so just because this zone is bigger does not mean that it's a more effective drug. This zone is larger than this zone, and you'll see that the indicators are roughly the same size. So this one actually probably would be a better drug to use over the cefotaximine drug that is also sensitive. I know that was kind of long-winded, but it does give you an idea of how you read those. Here is another one. This right here is a qualitative test. It doesn't really give you a number. It tells you susceptibility or resistance, but it doesn't give you what is a diagnosing an effective dose. And so you kind of are at a loss a bit as far as that. You just know if it's sensitive or resistant. If we come over here at this thing called the E-test, you'll see that this is a lot like the antibiotic discs here, but it is a long strip, and each one of these numbers is an increasing amount of the antibiotic on this strip. And each of these will be placed on a plate just like these discs are, and they will then go and you will have the antibiotics leaching out into the media. It will diffuse out into the media, and you will then have these bacteria that will grow up along here, and they kind of form this teardrop. Where this teardrop intersects the disc, that tells you the lowest dose that is usable and effective against this organism. So for this one, whatever IP is, I think it's a omeprazole, its lowest dose is going to be 1 microgram per milliliter. Okay, if we come over here at TZ, and I'm sorry I don't know what TZ is, you'll see here that its dose down here meets, and you'll see that you kind of have a faded area. It's not nearly as distinct as this. That means that this has some residual resistance here. So here, this is actually, its lowest dose is probably, I would probably say 1.5, or maybe between 1.5 and 1.0. So I do have a dosing number. Where I just know that my zone of inhibition is over 22 millimeters. I know it's at 27 millimeters, but what does that calculate to in grams or micrograms per milliliter? Here is the susceptibility chart for the Kirby-Bauer test. If you look here, you'll see that anything greater than 13 is susceptible, anything less than 8 is resistant, anything between there is going to be intermediate. If you look at these results here, you can see that your susceptibility, intermediate susceptibility, and resistance are going to be valued based on these numbers. When we talk about the minimum inhibitory concentration and the therapeutic index, these are ways that we can determine and look at how dosing is going to be effective. The minimum inhibitory concentration is the smallest concentration of a drug that will visibly inhibit the growth of bacteria. What is the highest dilution that it takes to cause this drug to begin to be diluted? Or to be, inhibited. Once therapy has begun, it is important to observe what type of response this patient is going to have. What is its clinical response? The smaller the MIC, the better, or the less likely it is to be toxic. If you look here, this is basically how you can determine a minimum inhibitory concentration. You start with your control. It's a negative control. It has no antibiotic in it. It only has bacteria. So we know our bacteria grows. Each next tube is going to increase the amount of drug by, it's going to double. And wherever you see visible inhibition, that is your minimum inhibitory concentration. So if we look here, we see that we have growth all the way up to 2 micrograms per milliliter. And at 4, we see visible inhibition. So the minimum inhibitory concentration of this drug against this organism would be 2 micrograms per milliliter. Okay? If we look down here at this picture, we have our control and we have our amount of drug. You'll see that we see visible inhibition here at 6.4 micrograms per, or microliters, and then at 3.2 microliters, we see that we still have a cloudy suspension. So this would be the minimum inhibitory concentration here would be 3.2 microliters for this particular drug against this particular organism. Now each drug is going to interact with each organism differently, and the MICs will be different accordingly. If treatment fails, it's going to be due to the inability of a drug to diffuse into a body part compartment where it needs to get. You have a few resistant cells that are going to continue to keep this ailment or this infection going, and it is not going to, they are not going to be sensitive to the antibiotic. And then the infection is going to be caused by more than one pathogen. You're going to have some which may be resistant to the drug. To choose the best drug, you have something called the therapeutic index. The best choice of the drug is going to be the one that has the highest selective toxicity. Means that it's going to be lower to the human, it's going to have a lower toxic potential to the human. So when we talk about therapeutic index, this is the ratio of a dose of a drug that is toxic to humans as compared to its minimum effective dose. Essentially, the smaller the ratio, the greater the potential for the drug interaction. Meaning that if I have a greater therapeutic index between where I begin to be toxic and where my drug is effective, that bigger index means that this is a better drug for me. If I have a narrow therapeutic index where my effective dose is one thing and where I begin to see a toxic effect is just a few ratios above that, then the potential for toxicity is much greater. So you want a big therapeutic index. Okay, this is the end of chapter 12. I will get this up and ready to you, and we will discuss any of these slides or any of these drugs that you are having trouble with. Be sure that you just come to class with your questions, and we'll go from there. Our next chapter is going to be chapter 13, and that is going to be the microbe-human interactions. This is where we're going to start talking about the immune system. Chapters 13 and 14 are the last two chapters of this section. I'm sorry, also 15. And I know that sounds like a lot, but it actually is not too bad because these are very short chapters. So 13, 14, and 15 will all be up here pretty quickly, and you will be able to get on them, and we'll go from there. All right, talk to you later. Bye.